Okay, we've had questions about how to make these fly dishes that we can dissect the flies on and record neural activity across the brain by stimulating segmental nerves and then recording in a muscle on the contralateral side, for example, or at least knowing through the circuit goes through the brain. So let me show you about these little dishes first of all. The glass part, you can buy that from Fisher or BWR. There's the size, 75 by 50. There are just these glass plates that you get, a box. And then the little magnetic strips are these business card magnetic strips. We get it at Office Max or our Business Supply House. What's nice about them is they have this peeling back that you can peel off. So cut your hole first in the center. It's really better to cut a hole in the center first, and then go ahead and then stick it onto the glass. And then you can trim the sides off if you don't get it exactly right. Just trim them off with the scalpel. And now for these little pins, if you can see these, there's actually an organization to them. If you notice that the pins are actually bent inwards, and you can see that, let's see if I can grab this one here. You can see that that is magnetic, and it's easy to move it back and forth, and you can pick it up. The problem is picking it up, it sometimes likes to flip on you like that. Or you can move it in the back, you know, over here from the back. You can grab it and move it around as you need. Now, we tend to bend these, and you can see this angle on this one, and angle them like that on both of them, going the other direction from the preparation. I find it easier just to move them around like that. And you can see then if your prep is in the middle, your pins don't get in the way. So you can move this back and forth relatively easy. It sticks. So let's look at the side of these things. If we put it on the side, you can see how it's bent. Let me see if I can get it on the side. There you go. And you see how it's bent here. The pin's bent and bent this way. So then it's easy to push pressure down on the cuticle. Alright, and what are these little things that are holding it? So the pins are insect pins, and that you can get from Biological Supply House or whatever. The relatively small ones. And then I took off clips off the back of vanilla, folders basically, the little tabs that you bend on the back of an envelope, and bent them, and then bent the wires the way I liked them, and kind of curled the wire around a little bit at the end. Put it on, put glue on, a little bit of super glue, then I put some epoxy on, and it stays really well. Why are they red? Well, to keep that metal from rusting, I went ahead and painted them with fingernail polish. Choose different colors if you want. You might have different colors for fixed dishes versus physiology dishes, and that way you don't mix them up if you're doing fixation. Okay, so that's the dish and the parts that you can get, relatively easy. Okay, well, in case you don't want to peel them off of envelopes, you can buy these bags of pins from Office Depot, Office Max. It just says 1.5 inch brass fitters. I just cut them with the wire cutter, and then bend them up, and then glue the wire to it, the little pins. After you pin them, you want to bend all your pins first, because if you glue it and then try to bend the pins, you probably pull the glue off the metal here. Okay, now if you want to know where these dishes were first described, they were first described in the Neurobiology of the Leech by Muller, Nichols, and Stent, the people that actually used to teach a leech course at Cold Spring Harbor and Woods Hole. And in the back of their book, let's find it here under Methods, Laboratory Manual, somewhere here. There it is. Actually, this first dish was described there, the arrangement for recording from leech ganglion preparations, and this was then modified to use for Drosophila preps. So if you're into leech neurobiology, which is really quite interesting, but at least we should cite the original source for these dishes. Some other people credited themselves for it, but actually it was first used in the leach system. So why the glass in the middle? Well, for the leach system, if you have a light that's coming through the bottom, then you could see the ganglion very well and all the neurons in the ganglion very well on a leach preparation, and that allowed them to poke individual cells that they were interested in, just like what we do now with the Drosophila, to poke individual muscle fibers. Okay? Okay, to make suction electrodes, to pull that in, I use this Chemex glass 34502. Pull it on a regular, puller. You can set the settings however you want for your pulling, and then I'll show you what we do with that next. So you can see these two halves have been pulled, and what we're going to do now is to break them off at different points, and sometimes they break really nice and sometimes they don't, and then we fire polish it down to get a nice fire polished tip. Okay, you can see the tips, and what we're going to do is to break them back like that, and then take the other one and break it back like that. And now I put them on a simple microscope with a little tungsten filament that we have bent here, a little holder that holds the glass, and then I just took some DC source and wired it up myself, a thing I found in the trash can, but it's got a differential source which we can turn on and raise the intensity of it to then fire polish the electrode. So you can see that one broke off too jagged, so I'm not going to use that one at all. Let's go look at the other one. The other half broke off very nicely. It's really a flat cut, a flat break, I guess you could say. And then you see the filament over to the left there, so what I'm going to do is turn on that and then fire polish the tip of this so I can even zoom in more. That tip diameter is about 5 microns, maybe a little bit bigger. You don't want it to touch the filament, it will stick, so I just keep it a slight distance away, and now it's going to start bending the tip of that a little bit, and it will be smooth. So I just let it sit there, and sometimes the filament moves because it heats up and bends a little. We can see that it's flared just a little bit there at the very tip. So it's heated up, and that's probably made it nice and smooth so it doesn't cut the nerve when you pull it in. Okay, so the setup is basically on the right hand side is a suction electrode that then goes connected to a stimulator. On the left side is the head stage, for an axle clamp 2B for intracellular recording. 2 wires, one the ground wire there, and the other wire is a ground wire for the stimulator. So basically we can stimulate, and if you want to see the setup, what we have here is the stimulator, an S88 grass, or now it's the company that bought them out, ADI Power Lab, and axle clamp 2B to record. We use filtering down here so we can filter the signal or amplify it there, and then it just goes on over to a computer. So after putting on the larvae, you might want to wash them a little bit, give them a bath. So that's saline that we use, and I'll put the formula of the saline. It's basically a modified HL3 saline. It's the same one that's in Clara DeCastro's paper. Let me see if I can get that back in there. There. And what's nice is that washes off the food that's on the animal from his food dish, and now we have a very clean larvae. And so we're going to go ahead and let me do a lower magnification here if I can, and we'll put them in a pin them down here. The scissors we use is from Fan Scientific Tools. The model item number is 15,000-10. It's a nice fine scissors. And then you really need like number 5 tweezers, but of course these number 5 tweezers all get damaged very quickly by students and people not being too careful. sometimes because they might use them for other things. So we put one pin right by the mouth hooks. I missed it that time with that pin because of this microscope, but there we go. Maybe that'll stay. Well, he's a fighter. There we go. Okay, so we got one pin in right behind the mouth hooks, and you can see the mouth hooks. And you can tell which side is the back of the Drosophila, right, because of the trachea that's running down the back. And then the second pin we'll put right behind tail of this animal. Well, right there. You just got to make sure not to push down on him or her because you don't want to damage the muscles. All right, let me put that back in view for you. There you go. See if we can clarify that a little bit. And now you can see the trachea running down the back of the animal on both sides. We could move this pin over just a hair because it's a little bit far back. And there's the problems when the pins flip on you. You're seeing all the issues. There we go. Hopefully we didn't damage the brain on that. And stretch them out a little bit. And if we look really close, let me zoom in again. And you can see the heart beating in there. There you go. So you can see the heart beating across here. We're going to cut right through here. So what we're going to do is make a cross section, just cutting very shallow in the cuticle here. Turn our scissors around and cut straight on down, right through the heart, all the way down between the two trachea. And then we're going to cut to the side a little bit over here, and that will allow us to put this other pin that we want to pull it out. And then we'll turn this prep around, and we will cut up over by the head, and we can go off to the left of this pin, and that'll give us two flaps that we can use. Okay, so let's get started. So the biggest problem I think are the guts coming out and then obscuring your view. You want to put your saline around the prep because that helps suspend the guts and everything and doesn't allow it to flatten out after you make this first cut. So I make the first cut. There's the mouth hooks up here. Here's our pin. I make it just a little, probably right over the brain basically. And there's our first cut. And now we have a little bit of GI tract bulging out. So now I'm going to turn the scissors sideways and find the little starting spot. There it is. And if you can, it's better to leave the one blade underneath the skin all the time. There we go. And we can cut right down past the trachea up there. Just cut that trachea off, we don't need it. So that's all opened up, as you can see. And this is some of the guts coming out, salivary gland. Now I'm going to turn it around and cut up towards the head because I'm right-handed. It's easier for me to do it this way. I know the light's shining off of it a little bit. Let's see if we can focus so you can see. There you go. And if I move the light to the side a little bit, now you can kind of see the guts that are coming out on the preparation, and that can be a little problematic. So let me put the light back so I can cut this. And what we're going to do is to push these guts down a little bit, get our blade underneath there, and cut again, and go right past up to the mouth hooks. There we go. Oops, now it came loose. That's okay, because what we can do now is grab our pin and pull it back out. Okay, and now I'm going to get our other pin ready to fillet this, and pulling it out, stretch it open. So let me see if I can get this turned around so I can grab the pin. I think the problem is really these pins sometimes, if you don't pick them up right, they flip on you and really causes some problems. So there we go. And if I pick it up past that little bend, it helps. So there, this one side. Let's see if we can re-pick this up, this other side, and stretch out the head region. So I'm going to pick this up. I'm going to try to grab the little flap there. There we go. Whoops, too much pulling. Pull the other side apart. There we go. Oops. So I'm showing you all the problems. I'm not cutting it out because it is a little difficult. Let me try to get this again. There we go. But you do want to be careful not to pull it too hard because then you pull on the muscles. Alright, so now we're going to go down to the tail region, or I call it the tail. It's really the caudal region. There's that one. And then we're going to pick this one up and kind of grab that flap if we can. There we go. Now we have a nice filleted prep. Let me put it in focus for you here. There's this prep. And we have all this GI tract now inside. So what we're going to try to do is to move some of it aside without going deep by grabbing any segmental nerves. We're just going to push it to the side like that. Okay. And now you can see some of that's to the side. There's a little bit more down here. I think I can just push that up. There we go. You can see the reproductive tissue here. One can actually sex the larvae being male or female by looking at these gonads and looking at the size. You can look at that online, figure out which one's which. And then we just pull some of that salivary gland and stuff out of the way, GI tract. There we go. Now, on the head region up here, you see this little bulb that's up there. That's actually the stomach, and sometimes that gets confused with the brain, some of the students. So if you pull this to the side, you can see the brain is underneath that, okay? But we don't want to just rip it out because it goes right through the central nervous system. So what we're going to do is just put it to the side and then take our scissors and cut that. And you have to be careful not to pull on the brain or cut the lobes of the brain. So what we have here is scissors. Can kind of push it up a little bit, get them in there and push it up, and then make the cut. Push it up and then make the cut. There we go. Now that's gone. The brain is underneath there. You can actually see it. These little trachea sometimes can cause problems because they push on your intercellular electrode when you're trying to reach over here to stab a particular muscle. So what I do is I cut them right there and there. And sometimes it's not, well, here it is. Sometimes the GI tract is really stuck to the anus region. Oh, shit. Okay. You see how I hit the pin? Caused a problem. There we go. Didn't want to stay. There we go. Try not to do that again. I hit the back of the little metal tab with my finger. Okay, so now what I'm going to try to do, I guess it's a little too bright maybe for you, is to take these scissors, and if you have the GI tract stuck to the anus, you don't want to just pull it because you'll pull your prep off your pins, almost like I did there earlier. And now we have these trachea here. What I tend to do is get one of the blades underneath there and kind of pull up on it. Make it close to the trachea so you don't grab any of the segmental nerves. There, that one's gone. Let's see if we can get this one from the side here. There we go. And if it wants to break off, you can always cut the, if you raise it up, you can cut them off. Let's see if we can grab this with the scissors, or we can go get the tweezers. There we go. Okay, so now we have everything pretty well cleaned up here out of the prep, except for this chunk of fat. Okay, now let's clean it all off, because if you put your intracellular electrode around there, you know what happens. These things will find the tip of that electrode and stick to it. And so what we do is use your tissue paper. Don't rub it across your prep of your larvae. It'll damage the muscle fibers. But I clean it all out. Put more saline on there, kind of really flood it so the tissue will stay floating. And now this is an important spot, so I'm going to zoom in a little bit and see if we can keep this. So what you can see here is maybe there's a little bit of tissue stuck up there by the brain. It's okay. We can get some of it off still if we wanted to. We can leave it alone too. We got two lobes down in there. I'll tell you what we'll do. We'll get a little bit more of that fat body out of there. Our stomach, whatever it was, GI tract. There we go. Let's pick up on it. There we go. We got that removed now. Okay, so you can see the two lobes. You can see the ventral nerve cord right here. There's the ventral nerve cord, and the segmental nerves, depending on what microscope you're looking at, you'll be able to see those relatively easy going off to the segments. To be able to stimulate a segmental nerve to go across the brain, or to look at some connections inside of there, like what we'll do is to cut, what I tend to do is just cut these lower segmental nerves by going like that. And maybe we can cut the second to the last segment, and you might damage the muscle fibers at this point down there. That's okay, because really we don't want them contracting when we stimulate. It just pulls on the prep. So now we have a bunch of these segmental nerves cut, and we can put it under the other microscope and pull those into a suction electrode. So for electrophysiology people, when you put your suction electrode in here and you'll see these segmental nerves, what you can do is to kind of have your suction electrode right here, pull a little vacuum, that'll pull the segmental nerves to the side. You can suck them up and then move your electrode on up a little bit, maybe what side you don't want to stab your muscle fibers, so your electrode will be sitting over here with these segmental nerves in it. If you want more segmental nerves, you can try to go along and cut down this edge. The more segmental nerves you get in there, though, it's probably going to recruit some motor neurons on the other side relatively easier, but you're more likely going to be damaging these muscle fibers trying to cut those nerves. When you make a cut there, try not to pull down on the segmental nerve. You want to cut and kind of pull up when you make the cut, so you're not pulling on the brain. If you pull on any of these segmental nerves from the ventral nerve cord, they seem to be damaged. So when you make those cuts, pull up a little bit like this. So you make a cut and pull, because we're not interested in this side. We want to then stimulate those segmental nerves going up. And then these muscles over here will be healthy to stab, particularly muscle 6, muscle 7. And in these segments up here, like in segment 2 and 3, they won't contract that much, or the whole prep won't contract that much, because you've cut these muscle fibers. So even though you're recruiting pretty many of all the motor neurons in these segments, it won't pop your electrode out too likely. But yeah, that happens too. So what you have to do is to remember to keep this tight so you don't have as much contraction. You can just touch these pins and kind of keep your prep taut. Okay? And that's about it for that. And then you stab your muscle fibers. I'll go see if we can get a picture of the electrode set up and suction electrode with some segmental nerves, and we can see the muscle fibers. Okay, so here's the brain and that preparation and the microscope that we just worked with, and some of the segmental nerves that are cut. I will, if I go down here and blow on the prep, you'll see them wiggling around actually. Okay, well, we'll pull them into a suction electrode. There they are. They're over to the side. You can see them over here to the side. They're over there. Okay, so let me go pull that in. Okay, so now you can see the nerves are pulled in there, and one could stab muscle 6 over to the right, or muscle 7 over there, and see if we get any activity. So you see we just got the nerves that were going down to the tip of the prep, the ones that we cut. And you can see on the left, there's still the segmental nerves that go to the muscle fibers we didn't cut. So if I blow that out, for example, there they are, and you see that the segmental nerves that we got. If you need to suck up more, you can. Do not pull too hard on the nerves because you will damage them. So you can even keep them a little bit away from the ventral nerve cord. Problem is you don't want to stimulate maybe the other ones that you're recording from. But you can see if you get these two tail nerves, I call them tail nerves, the caudal nerves, go to the last segments down there, then any motor neuron that you record from, or I mean muscle fiber, then you know you're going through the CNS. Or you could just get the ones on the contralateral side if you want to. So now it's very easy to put an intracellular electrode and stab muscle 6 right over there to the right hand side, the nice wide muscle fiber that you see. Let's see if we can zoom in on it. Yeah, there it is. Muscles 7, 6. The whole lineup is nicely laid out there. And we can then stimulate that. I'll blow those nerves out again. There you go. Okay, and then standard electrophysiology.